Hello, my name is Jennifer Hiscock and I'm a lecturer in the School of Physical Sciences here at the University of Kent. I am a supramolecular chemist. What does that mean? That means I study non-covalent interactions between molecules. What are non-covalent interactions? Well, the definition's in the word. Anything that doesn't involve a covalent bond. So what can we consider under this non-covalent interactions title? Ion-ion interactions, ion-dipole interactions, dipole-dipole interactions, and pi-pi interactions, and van der Waals forces. In particular, I have a little bit of an interest in a specific form of dipole-dipole interaction, the hydrogen bond. So, non-covalent interactions, you can think of them as molecular Velcro. Molecules can come together, fall apart, come together, fall apart, come together, fall apart, with no damage at all to that molecule, just through the formation of these reversible interactions. But why do we care about how molecules come together, how they fall apart, how they can aggregate? Well, this principle underpins soft material development, such as the hair gels that you buy at your local supermarket, the medicinal surface gels you put on your arm for pain relief. So development and understanding of these materials is very important. We can also look at them for new drug development. We all know today that the development of novel antimicrobials and antibiotics is really important as antimicrobial resistance to our drugs that we normally take to fight infections increases. So if we can use these associative interactions to look at the formulation of new drugs, we're on to a winner. We can also use these molecular self-associations to produce novel capsules to carry drugs to a specific site within the human body. Therefore, the drugs don't come in contact with the entire system, just to a specific site. And we can also look at these interactions within surfactant systems. What are surfactants? Why are they important? Well, you use soap every day, washing up your dishes and shampooing your hair. And also, it's a big thing in the automotive industry for removing and breaking down oils. We can also look at these associative interactions to produce novel catalysts. Why do we want to produce novel catalysts? Well, we all want to decrease the amount of energy it takes for chemical reactions to take place. When you do these chemical reactions on huge scales, it can take a lot of energy to drive them to completion. Catalysts reduce this energy. So using these molecular interactions to bring specific parts of a molecular system together to produce effective catalysts is a good way forwards. But how do we study molecular interactions? Molecules are really small. Well, the first way perhaps is looking in the solid state. So we can take a solution of your molecules, such as shown here, and we can allow this solution to evaporate. When the solutions evaporate, you can produce crystals. You can use a microscope to look at these crystals and see if they're single in nature. If they're single in nature, then we can use a single crystal X-ray diffractometer to mount these crystals, fire X-rays at them. The X-rays then diffract off the crystals and are collected by the detector. The detector can then produce information that through some complex mathematics allows us to see how these molecules interact within the crystal structure. This is one of our compounds. We can see that it forms a dimer. Two molecules come together through the formation of 4 hydrogen bonds. The NHs act as the hydrogen bond donor, and the oxygens attached to the sulfur act as the hydrogen bond acceptors. So now we know that this particular type of molecule can come together and form dimers through the formation of 4 hydrogen bonds. But if we're going to look at the production of novel drug carriers, bits and pieces like that, I don't know about you, but I'm mostly liquid. So we can't really rely on solid state studies. We have to look into the solution state. What can we use to do this? Well, we can use NMR, okay? Nuclear magnetic resonance spectroscopy. And what we can also see in the spectra shown behind me is a proton NMR. This proton NMR, the different signals correspond to the different proton environments within our molecule. So each different spike is a different proton on our molecular structure. Now if we layer some of these proton experiments together, again do some really funky maths with it, we can produce a DOSY experiment, diffusion ordered spectroscopy. What's the importance of this? Well, it looks at how quickly things diffuse through the solution. Big things diffuse slowly, small things diffuse really fast. From these diffusion rates, we can calculate the size of the molecular species. If we have 2 molecules stuck together, their size is going to be quite small. If we have 10 molecules stuck together, it's going to be bigger. 20, even bigger. 1,000, massive. And this is going to be something we can see using this technique. So, from the spectra shown behind me, basically indicates that our molecules exist either as monomers or dimers in the solution, which is exactly what we expect from the solid state experiments. But do we have hydrogen bonding? Are these monomers held together by the formation of hydrogen bonds, and how strong are they? Well, we can use another really simple proton experiment. We can take a solution of our compound and we can look at the signals that correspond to the hydrogen bond donors, those NH groups. And as we dilute our sample, we can look at how those signals change. Plotting those signals and doing some more funky mathematics, we can calculate the strength of that molecular interaction. So now we know that these molecules come together, probably as dimers in the solution state, and how strong those interactions are. But what does this mean about global properties? Okay, how do these molecules affect the solutions that they're in? Well, we can observe surface tension using a tensiometer, such as the one shown behind me. This machine basically looks at a droplet that's produced. This droplet contains some of our compound. And from the shape of that droplet, we can calculate the surface tension. So, at a low concentration, with not very much of our compound within the solution, we see a high surface tension. We increase the concentration of our compound and we start to decrease the surface tension. But why is this happening? Well, this is because all of our little molecules are aggregating at the surface of that droplet. This lowers the surface tension. So we add more and more compound, we lower the surface tension. But it gets to a point, the surface tension doesn't lower anymore. We keep adding compound, we just get a flat line. Why is this? Well, it's because the surface of that droplet is saturated. There is literally no more room to aggregate any molecules at the surface. So they have to stay inside. But if they're staying inside, what are they doing? They can't be aggregating at the surface, so they've got to be doing something. Cue the fact these molecules are intrinsically fluorescent. What does that mean? Well, this means that we can use microscopy techniques to look at the big structures, not the little ones, the big ones that exist in solution. Here we have an image of the structures formed in DMSO. You can see that they look fairly solid, they're circular in size, and they, to be honest, to me, don't look too interesting. Here, on the other hand, is what happens when we have a solution that's mostly water. This is 95% water and only 5% DMSO. What do we have? We have a hollow bubble with a fluorescent edge to it. What does this mean? This means all of our fluorescent molecules are aggregated at the edge of that bubble. So our molecules are coming together and interacting to form this hollow structure. And that's pretty cool. We can start to put things inside this and we can start to use it to do more interesting things. But we want to be able to control the formation of these structures in solution. So we start to look for trends. So here's some information from 5 of our compounds. And we can see that as we increase the dimerization constant, so as the strength of the interaction between those molecules when they come together increases, we get a decrease in critical micelle concentration. So this critical micelle concentration is that turning point that we saw on the graph earlier, where you no longer get a decrease in surface tension, but you get that flat line. So just where those two things meet is our critical micelle concentration. But this is pretty cool, because if I want to get a high yield of my circular hollow species in solution, what do I need to do? Well, I now have some information that allows me to design a molecule that does that better. I increase the strength of that dimerization. I get a lower critical micelle concentration, and therefore more of my molecules can be used to form the spherical species in solution. So if you'd like to learn any more about this and look at it in a bit more detail, if you just follow the DOI from this chemical science paper shown behind me, it'll give you all the information you need. Thank you very much for listening.